Welcome to The Explainer. Today, we're taking a fascinating yet really grounded and measured journey into the very foundation of modern molecular biology. We're going to explore what is honestly a massive paradigm shift in healthcare and clinical research. We're talking about the transition from classical biochemistry to the modern multi-omics revolution. Now, what does that actually mean? Well, we'll be looking closely at how scientists are no longer just examining a single molecule in isolation. Instead, they're capturing the entire unbelievably complex molecular universe operating inside of us right at this very second. It's a shift that fundamentally dictates how we understand human health and disease. So let's dive right in.
Here is our roadmap for today's explainer. First, precision medicine. Second, the omics cascade. Third, classical versus modern approaches. Fourth, experimental workflow. Fifth, proteomic complexity and mass spectrometry. And finally, number 6, inborn errors of metabolism.
Section 1, Precision Medicine, Beyond One Size Fits All. Now, please don't worry if those terms on our agenda sounded a bit dense. We're going to break these biochemical concepts down methodically, step by step. Let's start with our very first major concept, which is precision medicine. For the longest time, medicine has been dominated by a sort of generalized philosophy. But thankfully, we are moving well beyond the era of broad, sweeping treatments. Just think about the standard approach to medicine, the old one-size-fits-all strategy. In classical medicine, you might have a really diverse population of patients, all suffering from what looks like the same disease. So the standard approach gives every single one of them the exact same drug, just hoping it works. But as we know, a single drug might only be effective for a tiny fraction of those people. Precision medicine flips that on its head. By integrating multi-omics data, we can actually group patients based on their unique molecular biomarkers. For instance, instead of just giving standard aggressive chemotherapy to all breast cancer patients across the board, precision medicine allows us to look for specific molecular biomarkers, like, say, the overexpression of the HER2 protein. Once we find that, we can apply targeted therapies that directly inhibit that specific protein. It's incredibly precise, and it vastly improves individual patient outcomes.
Section 2, the omics cascade, mapping biological information.
To really grasp how we gather this incredibly detailed molecular profile, we have to map out the flow of biological information. You can kind of think of this as the central dogma of biology, but expanded for the modern omics era. At the very beginning, the top level, we have genomics. This analyzes our DNA. And genomics basically tells us what can happen. It is the architectural blueprint of you. Next on the line is transcriptomics, which analyzes RNA. You can think of these as the intermediary messages being sent down to the cellular factory floor. It tells us what is about to happen. Then that RNA is translated into proteins, which is studied by proteomics. Proteins are the functional, physical machinery of the cell, so they tell us what is happening. And finally, at the very end of the line, we reach metabolomics. This is the study of small molecules and metabolites. Because this represents the final end products of all those cellular processes we just mentioned, metabolomics tells us exactly what has happened.
I want to pause here for a second because this concept is absolutely crucial. The metabolome detects the picture that is more clear and more near to the phenotype of that precise moment. It's a profound way to look at biology. Think about it like this. Your genome dictates everything about you, sure, but it really only shows potential. You might carry a genetic mutation that's linked to a specific type of cancer. That genomic data tells us cancer is a distinct possibility, but it absolutely does not guarantee that you have cancer right now. However, if we look at your metabolome and we see a specific disease biomarker that is actively elevated, well, that provides a real-time, undeniable snapshot. It proves that a pathological condition is actively ongoing in your body right at this very second. So let's firmly define what the metabolome actually is. It's the total complement of all low molecular weight metabolites in a biological system. We're talking about basic amino acids, simple sugars, and lipids floating around in a cell, a tissue, or an entire organism. It represents a truly comprehensive picture of all the metabolic processes that have occurred or are currently occurring. By analyzing the metabolome, scientists get the ultimate, dynamic measure of an organism's pathophysiological changes. Long story short, it is the absolute closest molecular reflection of your current state of health.
Now, nested right inside metabolomics, there is an incredibly important independent branch called lipidomics. Usually, metabolites are distinguished by their polarity, and lipids form this massive class of mostly hydrophobic, meaning they repel water, small molecules. And it's important to note they aren't just stored fat waiting to be burned for energy. There is a tightly coordinated set of lipid anabolism and catabolism, basically the biological building up and breaking down of these molecules. This delicate balance is crucial for energy homeostasis, the literal physical structure of our cell membranes, and vital signaling processes. When your lipidome is imbalanced, it directly contributes to various human diseases, which makes lipids vital biomarkers to track.
Section 3. The evolution of the toolbox. Classical versus modern. All right, now that we know what molecular information we're actually looking for, how do we find it? Conceptually, the difference between classical laboratory methods and modern omics is just staggering. In classical biochemistry, you typically run an analysis to measure or quantify a single already known molecule. For instance, your doctor might order a routine blood test to look at just one specific hepatic enzyme, like ALT or AST, just to check on your liver function. But measuring just one enzyme can only tell you about its specific isolated role. Modern omics, on the other hand, investigates massive, comprehensive sets of molecules all at the exact same time. We aren't just checking your ALT or AST levels. We are looking at the entire complex biochemical environment that those enzymes operate within. There's a great analogy that perfectly captures this difference in scale. Classical biochemistry is a lot like a scientist sitting on a dock with a single fishing pole. They know exactly what fish they want to catch, say that one specific liver enzyme, and they spend all their time carefully trying to hook that one specific catch. It's very precise, sure, but it's inherently limited. Modern omics is completely different. It's like bringing a massive industrial crane and a huge trawling net out to the ocean. We don't just want one fish. We want to scoop up the entire biological ecosystem at once, so we can see exactly what is living and interacting together in that water.
Section 4. Design and experimental workflow.
So, practically speaking, how do scientists actually operate this massive molecular trawling net in a lab? Well, the experimental workflow is highly systematic. Step 1. Scientists have to choose a biological matrix. Blood is obviously the classic choice here, but what's truly fascinating right now is the shift toward non-invasive matrices, like urine and especially saliva. Saliva is becoming a real game changer. It's easy to collect, it's cost-effective, and most importantly, it causes significantly less stress for patients who need constant routine monitoring. Step 2 involves extracting and preparing the sample, which often means digesting larger proteins down into smaller, manageable peptides. Step 3 is data acquisition, which relies heavily on mass spectrometry. And finally, step 4 is data processing. This is where advanced statistical analysis combs through massive, mind-boggling datasets to find those relevant biomarkers.
Within this workflow, researchers have to make a choice. Are they using an untargeted or a targeted approach? You'd use an untargeted approach when you're in the discovery phase. You cast a super wide net without a specific hypothesis, just to see what molecules are present out of thousands of possibilities. This gives you what's called a relative quantification, which basically just means knowing you have more or less of a certain molecule than you did before. On the flip side, if you already know the specific biomarkers you want to monitor, you use a targeted approach. This focuses on a smaller specific subset of known molecules, and it allows for absolute quantification. That means knowing exactly how many milligrams or picograms of that specific molecule are actively present in your sample. Section 5. The challenges of the proteome. While that workflow might sound relatively straightforward, the biological reality of studying proteins is anything but simple. The proteome presents 3 massive hurdles for researchers. First is variability. The proteome is highly individual-dependent, time-dependent, and even environment-dependent. So, finding a universal biomarker that holds true across everyone? Incredibly difficult. Second is dynamicity. In a single sample of blood, some proteins are present in huge, overwhelming quantities, like albumin. Meanwhile, vital signaling proteins might be present in just a handful of copies. Trying to detect these ultra-low abundance proteins hiding among the giants is quite literally like trying to clearly record a whisper at a heavy metal rock concert. It's tough. And finally, there is immense complexity. Let's talk about that complexity for a second. A single human gene doesn't just make one simple protein and call it a day. Through a process called alternative splicing, that one single gene can actually create multiple different protein isoforms. And it gets crazier. It doesn't stop there. Those isoforms can undergo what we call post-translational modifications. You can think of these modifications as little chemical tags, like a phosphate group or a sugar molecule that get added to the protein after it's already built. It completely changes how the protein behaves and functions. Each unique combination creates a distinct proteoform. Because of this whole process, the roughly 20,000 genes in our human DNA can exponentially generate over 1 million fundamentally different proteoforms in the body. It's a massive puzzle. Section 6. Mass spectrometry. the engine of omics. So to successfully navigate this incredible molecular complexity, scientists have to rely on some unbelievably sophisticated hardware, specifically the triple quadrupole mass spectrometer. It acts kind of like a highly advanced molecular weigh station. Here's how it works. Molecules are injected and directed to the first quadrupole, which carefully measures the exact mass of the intact precursor molecule. Next, the molecule enters the second quadrupole. This acts as a collision cell, literally shattering the molecule into pieces. Now, why on earth would we shatter it? Because the specific weights of those shattered fragments act like an undeniable, unique fingerprint for that exact molecule. Finally, these pieces pass into the third quadrupole, which measures the masses of all those resulting fragments. By matching this unique fingerprint against a theoretical database, scientists can definitively identify the precise metabolite they're looking at.
Section 7. Clinical Applications. Inborn Errors of Metabolism. Okay, we have covered a lot of heavy theory and methodology today. Let's ground all of this in tangible human reality by looking at some specific clinical applications. We're going to focus on inborn errors of metabolism, or IEMs. These are congenital genetic conditions, meaning they are present at birth, that are fundamentally driven by an enzymatic deficiency. To make sense of this, think of a standard metabolic pathway in your body like an industrial assembly line. If the specific enzyme responsible for step B on that line is broken, or just completely missing, the entire assembly line comes to a screeching halt. The physiological pathway is disrupted, and that leads to a profound metabolic disorder. So what exactly happens when that biological assembly line halts? Well, because the enzyme isn't working, it can't process its substrate. As a result, that substrate massively and dangerously accumulates in the body, eventually causing direct toxicity. A really classic example of this is phenylketonuria, or PKU. In PKU, an enzyme deficiency causes the amino acid phenylalanine to accumulate, which, if left unchecked, can lead to severe brain damage. But today, thanks to targeted omics, a simple heel prick blood test on a newborn uses mass spectrometry to find that elevated substrate instantly. It allows doctors to intervene with a specific diet before any damage ever occurs. We see similar mechanisms in type 1 diabetes, where a lack of insulin causes glucose to dangerously accumulate. And beyond that, multi-omics is also allowing for the identification of early cancer markers, detecting minute metabolic shifts long before a tumor is ever visible on a traditional scan. We've covered a lot of ground today. We've seen how precision medicine uses the omics cascade to move us far beyond generalized one-size-fits-all treatments. We've contrasted classical single-molecule tests with the massive trawling nets of modern omics. We explored the immense million-piece puzzle of the proteome, and we've seen how mass spectrometry acts as the engine to uncover life-saving biomarkers for metabolic diseases like PKU. So, as we wrap up, I want to leave you with this final provocative thought. If our metabolome truly provides a real-time, highly detailed, and accurate snapshot of our ongoing health, how soon will your routine yearly physical blood test be entirely replaced by comprehensive, individualized, multi-omic profiling? It's a fascinating future to think about. Thank you so much for joining me on this explainer. Keep questioning and keep learning.